All right, let's just dive right into it. Welcome to the Explainer. Today we're looking at this absolutely fascinating collision between textbook biochemistry and gritty real-world pathology. We are talking about your body's metabolic waste, those microscopic filtration plants working nonstop inside you, and what actually happens when rogue bacteria completely hijack the whole system.
Seriously, think about this. How does a tiny, swarming bacteria actually manage to turn your urine into solid rock? I know, it sounds straight out of science fiction, right? But it's a very severe clinical reality called an infection stone. To really grasp how a microscopic pathogen can engineer an actual geological formation inside your kidney, well, we've got to understand the biological foundations of waste first. So I'm going to hand it over to host one to lay down the ground rules.
Thanks. Okay, so here's our roadmap for today. First, nitrogenous wastes and habitats. Then, we hit the urea cycle blueprint, followed by the anatomy of excretion. After that, we'll get into infections, stones, and pathogens, tackle some clinical disorders, and wrap it all up with our 10-second summary.
Let's kick things off with Section 1, Nitrogenous Wastes and Habitats.
So when your body breaks down or catabolizes proteins and nucleic acids, it strips away the useful stuff, the carbon, hydrogen, and oxygen. But what's left over? Nitrogen. And excess nitrogen quickly forms ammonia. You really have to think of ammonia as your body's toxic exhaust fumes. It raises the pH of your body fluids to incredibly dangerous levels, so it absolutely needs a chemical detox plant to be safely packaged up and removed. Now, how do different animals handle all this toxic exhaust? Well, aquatic animals are ammonotelic. They just excrete pure ammonia directly out into the water. It's a strategy that requires zero energy, but it takes huge amounts of water to dilute it safely. Terrestrial mammals, like us, we're ureotelic. We convert that ammonia into urea, which is way less toxic and really saves on water. And then you've got birds and reptiles. They're uricotelic. They excrete uric acid as this thick white paste. Now listen closely, because here is a classic board exam trap. Do not mix up the evolutionary trade-offs. Ammonotelic saves energy but wastes a ton of water. Uricotelic, on the flip side, saves the maximal amount of water but costs the absolute most ATP energy to synthesize.
Moving right along to Section 2, the Urea Cycle Blueprint.
Okay, so take a look at the Krebs-Henselt urea cycle. Discovered way back in 1932, this is basically your liver's main machine for converting that toxic ammonia we just talked about into safe urea. And what's really cool is how the cycle is physically split. The first part happens deep inside the mitochondria, but then the rest of it takes place out in the cytosol of the cell. For your exams, the absolute crucial point here is nailing down these 5 chronological milestones. Step 1, ammonia and carbon dioxide condensed to form carbomoyl phosphate. This is catalyzed by CPS1 in the mitochondria, and heads up, this step costs 2 ATP. Step 2, it combines with ornithine to form citrulline. Step 3, now we're out in the cytosol, and it condenses with aspartate to form arginosuccinate. Step 4, this gets cleaved to release fumarate, which, by the way, is a super neat connecting link right back to the Krebs cycle. And finally, Step 5, arginase cleaves arginine to form our ultimate target, urea, while regenerating ornithine to keep the whole cycle spinning. Actually, let me jump in here with a quick lab spotlight, because, memorizing the pathway is really only half the battle. Out in a clinical lab, we measure something called blood urea nitrogen, or BUN. Think about it. If the liver is making urea perfectly fine, but the BUN levels in the blood are suddenly spiking, what does that actually tell us? Well, it tells us the kidneys, which are supposed to be filtering this urea out, are failing. So we use BUN as this critical, independent diagnostic variable to assess real-world renal function.
Spot on. All right, let's get into Section 3, Anatomy of Excretion.
Let's zoom out and look at the functional unit of the kidney, the nephron. Blood rushes from the wide afferent arteriole straight into the glomerulus. That's a super dense capillary network packed inside Bowman's capsule. From there, the filtrate takes quite a journey. It flows through the proximal convoluted tubule, dips down the loop of Henle, travels up into the distal convoluted tubule, and finally dumps right into the collecting duct. Now, the physiology behind this happens in 3 very strict phases. First up is glomerular filtration, where high pressure literally forces water, urea, and small molecules out of the blood.
Second is selective reabsorption. This is where the body actively and passively grabs back all the good stuff it needs, like glucose and amino acids. Third is tubular secretion, which is the active pumping of excess hydrogen and potassium ions into the filtrate just to keep your blood pH perfectly balanced. Oh, and here's another huge exam trap for you. Excretion is the elimination of metabolic wastes like urea from your cells. Defecation, on the other hand, is just the removal of undigested food from your digestive tract. Do not confuse the two.
Okay, time for section 4, infection stones and pathogens.
Check out this intense contrast between the microscopic and the macroscopic. We've got an H&E stain of Proteus mirabilis. That's a gram-negative, rod-shaped bacterium completely famous for its swarming motility. And then contrast that with the gross pathology of a kidney completely obstructed by a calculus, with literally yellow pus oozing out. Proteus mirabilis is notorious for causing these stubborn, catheter-associated urinary tract infections. It builds these incredibly dense biofilms and pumps out cytotoxins that violently damage the lining of your bladder.
But how does a bacteria actually build a solid stone? Well, it all comes down to a single enzyme, urease. Proteus secretes urease, which rapidly hydrolyzes the urea that host 1 was just telling you about. It basically smashes urea apart into carbon dioxide and ammonia. And that liberated ammonia, it dramatically skyrockets the urine pH to a highly alkaline 7.2 to 8.0. This sudden alkaline shift makes normally soluble magnesium and phosphate just crash right out of the solution, precipitating into solid magnesium ammonium phosphate. And that is what we call a struvite stone.
Now, you might be wondering, how do we actually prove this exact mechanism in a research lab? We don't just guess, we use mutant control groups. Scientists will take wild-type Proteus mirabilis, the normal, fully functional bacteria, and compare it directly against mutant strains, like maybe a mutant that lacks flagella, or one missing the HPM-A gene for the hemolysin toxin. By isolating these independent variables, researchers found that, hey, without flagella, the bacteria literally cannot swim close enough to invade host cells unless they are artificially centrifuged onto them. This perfectly proves exactly which molecular tools the pathogen relies on to cause all this destruction.
Fascinating stuff. All right, section 5, clinical disorders.
Let's quickly break down the 3 main types of stones you absolutely need to memorize. First, calcium oxalate stones. These are by far the most common, making up about 75% of cases, and they typically form when urine pH is acidic, so under 7.2. Next, uric acid stones. These make up about 8%, also forming in acidic urine, and they're heavily linked to gout, which is where uric acid crystals painfully accumulate in the joints. Finally, we have struvite stones. They make up just 1% of cases, but as host 2 just brilliantly explained, they are exclusively linked to urease-producing bacterial infections, and they absolutely require an alkaline pH to form.
And that brings us to section 6, the 10-second summary. Okay, here are your absolute high-yield takeaways right before the exam. 1. Highly toxic ammonia is detoxified into urea in mammals and uric acid in birds. 2. The urea cycle relies on 5 strict enzyme steps that bridge the mitochondria and the cytosol. 3. Urine formation requires glomerular filtration, selective reabsorption, and tubular secretion. And 4. Proteus mirabilis uses the urease enzyme to alkalinize urine, precipitating those massive struvite infection stones.
Perfect. And I'm going to leave you with one final clinical mystery just to really test your understanding. If a newborn baby has a genetic mutation, an inborn error of metabolism, that completely blocks one of those proximal urea cycle enzymes like CPS1, what catastrophic event happens in their blood within the very first 24 hours of life? Think about what builds up when the body's detox plant completely shuts down. The answer is profound hyperammonemia, a rapid, incredibly toxic buildup of ammonia that directly targets the central nervous system. You see, biology isn't just pathways on a textbook page. It is a very delicate, real-world balance keeping us all alive. Thanks so much for tuning into The Explainer, and keep questioning the science.